My screen? Can you hear me?
Yes, sir, we can see.
Oh, screen दिख रहा है?
Yes, sir. Yes, sir. You can start.
Can you go ahead? Yeah. Good morning, ladies and gentlemen. This is Valiappan from Coimbatore. It's a pleasure talking to all of you this morning. I deem it an honor to associate myself with this webinar organized by College of Pharmaceutical Sciences, Dayananda Sagar University of Bangalore. Let me place on record my sincere thanks to my colleague, Professor Murugan, the Dean, College of Pharmaceutical Sciences, for this opportunity. I also thank Dr. Richa Sood, who has been coordinating with me for the conduct of the webinar. My thanks to Kalpana Divakar. I believe the principal, the Dean is not available today, so she has come in, and I thank her also for the nice words she has spoken about me. So this topic is of immense relevance from two perspectives. One is from the COVID pandemic, other one is from the explosion of chiral technology. And today, there's an exponential explosion of chiral technology and a lot of pressure from regulatory agencies for the production of enantiopior drugs, and also to have a mirror image perspective when performing pharmacokinetic, pharmacodynamic, and toxicological studies if the molecule under question is chiral. So this presentation has got two points of interest. One is from the perspective of COVID, and another one is from the perspective of chiral technology and a repurposing strategy. So understand the time slot is 60 minutes. I believe I should be able to get it done with that. With this note, let me get into my presentation, exploring chiral switches, a repurposing strategy for potential drugs to treat COVID-19. The focus will be on rationale, facts, and hopes. This is a disclaimer I wanted to put because I'm neither a microbiologist nor a microbiologist. COVID-19 it's a short form of coronavirus disease 2019. You find here the COVID-19. So that's what has come up. Formerly, this disease was referred to as 2019 novel coronavirus or 2019 nCoV. So this is the sketch of the presentation, and I plan to make the presentation in two sections. The first section will be on COVID-19 and overview. Will be spending around 15 to 20 minutes approximately, and you will have 5 minutes of video at the end of the first section. Then we'll get into the second part where we'll be talking about the need for repurposing strategy, the mirror image perspective, then the hopes, and finally the closing comments. So that will be, I'll be spending around 30 minutes, I believe. So in total, you'll be able to done within 60 minutes. So with this, let me get into an overview. So let's take a look at the history of COVID naming, especially during the 3 zoonotic outbreaks, in relation to the virus taxonomy and disease caused by these viruses. When I mean zoonotic outbreaks, I mean zoonotic virus is a virus which can jump from animals to human beings. Any virus which can jump from animal to human beings, you call them as a zoonotic virus, and the disease is called a zoonotic disease. And we had approximately recently last couple of from 2012 we have 3 zoonotic outbreaks. The first one is in 2003. That is called SARS, Severe Acute Respiratory Syndrome. The virus name is called SARS-CoV, and the ID is 65. And the virus species is called the Severe Acute Respiratory Syndrome related coronavirus. So the naming authorities there are two authorities here. One is the WHO, and the other one is the International Committee of Taxonomy on Viruses, and the Corona Study Group. One minute. Excuse me, committee. And in 2012 we have what is called as MERS, which is called Middle East Respiratory Syndrome, and it's a virus is MERS COVID, and the international coding today is ID64, and the virus species is named as Middle East Respiratory Syndrome related coronavirus. And today we have 2019 the COVID, then it's very closely related to SARS-CoV, but then it's named as SARS-CoV-2, and the virus, the species is Severe Acute Respiratory Syndrome related coronavirus. So this is the kind of history of COVID naming, and these are the species, types of human coronavirus responsible for health hazards. There are basically 7 strains. There are 4 genera, as we call them as alpha, beta, gamma, and delta. In the alpha, we got a couple of them which generally put you in the symptoms of common cold. It was, identified in 2004. In 1960, we had one alpha coronavirus symptom, same as a common cold. 1960, we had another one. But the interesting things have come up in 2002, the first SARS-CoV, the potential severe respiratory tract infections. Then we moved on to 2012, we had MERS, it's a Middle East respiratory, issue there, and this is a potentially severe respiratory tract infections. Then in 2020, we had the COVID-19, the SARS-CoV-2, potentially severe respiratory tract infection. So this is how, and there are basically 7 strains which are, and the gamma and delta are not known yet in human beings. So this is how the types of human coronavirus you classify them. Then let's just take a look at the WHO timeline and COVID-19. In November 2019, first China case of Wuhan, we had a report of coronavirus case. Then December 2019, first pneumonia case again in China. Then in December 31st, 2019, WHO reported to China about the coronavirus case, and then they tracked it to a vet market named seafood market Wuhan in China, where there is a chance of transmission of pathogen from wild animals to humans. This is called as a zoonotic virus. So the first identified case was from a vet market named Hunan Seafood Market in China. Then later in January 2020, first case in USA it happened, and then in March 2020, WHO announces it as a pandemic. Normally epidemic is contained within a country. When it crosses multiple countries, you call them as a pandemic, and Hindi you call it as a Mahamari. In March 2020, India locks down for 67 days, March 24th to May 1st. Then April 2020, India reports 430 cases. July 17th, 2020, India reports 10 lakhs. And as on today, September 17th, I think approximately crossing 51 lakh cases, and still we are not sure about it. So that's how the pandemic is, it's really creating problem for everyone. Let's take a look at the analysis of biological features of SARS-CoV-2. This is taken from the John Hopkinson's website, which is world over. People are referring to that. Corona Resource Center, and the emergence date of COVID-2 is December 2019. The area of emergency is Wuhan, China. The key host is bat. The number of countries infected is 188. The entry receptors in humans is angiotensin converting enzymes. A very interesting enzyme which we will be looking at a little later, AC2. This is where the virus connects first and then enters into the body. Signs and symptoms basically cough, fever, shortness, and breath. Disease is caused by SARS-CoV-19. So if you look at approximately some data here, total infected patients across the blue is in globe, across the world, black is in USA, India is in green, and this is in Tamil Nadu, because and Bangalore is very close to that. So total infected patients we have is 29.9 millions across the world, and 6.6 millions in USA, and in India it's crossing 51.1 lakh as of today, and in Tamil Nadu is approximately 5.1 lakhs. The total recovered patients here is 20.3 millions. Here it is 2.5 million in USA. India it is 40.3 lakhs, and Tamil Nadu it is somewhere around 4.58 lakhs. Total died patients 0.9 millions, and here it is somewhere around 0.19 million, and in India it is the patients died approximately is 83,000 and all. It is in Tamil Nadu, it will be around 8,000 or Bangalore is very close to that. The interesting thing we have to note here is the mortality rate. If you look at that across the globe, it is 3.1%. Mortality rate means total infected patients to the number of deaths happening, that ratio. If you look at globe, it is 3.1. US is somewhere around 3.01 here. And in India is doing much good there. We only have 1.63%, and Tamil Nadu is very close to that 1.6, and Bangalore is still a little lower than that, closely with it. So this just try to explore this mortality rate a little more. This is the mortality analysis of most affected countries in the world, and you find here India is at the bottom in this case. That's good. We are approximately, this is a scale of 10%. You find here it is less than 2%, 1.6% is the mortality, the ratio. That's observed case to death ratio. In US comes somewhere around 3%. There are countries which are crossing 10%. So I think in this aspect, we are able to contain the number of deaths, and that's a good news for us. Interesting, I just wanted to look at the number of publications happening, after the coronavirus, especially after the SARS-CoV 2002. This is data is from 2003. You're plotting the number of articles related to coronavirus, the years. You find that between 2004 to 2020, you find more than 1.13 million papers are published on coronavirus. And this happened, if you look at that, 2003 we had SARS, 2002 SARS-CoV virus, and after that there's an surge of interest in this coronavirus activities and a lot of publications. If you look at 2020, you will find that the number of publications have gone almost close to 40,000 publications exclusively on coronavirus. So it's the kind of research activities going on. It's an interesting thing. And this is the slide showing the transmission, how your virus gets into you. So it's the primary host is believed to be the bat. From the bat, the coronavirus gets into an intermediate host called as pangolin. Pangolin is a wild animal sold at the seafood market of Wuhan, China. This is an anteater, and in China people eat this. Possibly, it got into human beings from that. Then once you are infected, then you can get into another person by what is called as droplet infection. If you are within a distance of 2 meters or approximately 6 feet, that's why people say you maintain social distancing and you don't get much closer. Then other possible contamination is through transmission through fomite infections. I'll tell you what it is. Then it is aerosol. So these two are a little less susceptible, but the major transmission can happen by SARS virus droplet infection. Fomite, what happens is when it is like infected person touches a surface like a table or a mobile or a laptop or a car door, then another person touches that, he gets infected, and then you touch on your nose, eyes, and all that, then the person get infected. So this is called as fomite infection. Involves exposure to inanimate objects, could be clothes, utensils, furniture, which are likely to carry pathogen from one susceptible person to another during the touching surface. Then later followed by eyes, nose, or mouth, you get infected. So this is called fomite. That's why people say surface, don't touch it, because these viruses can stay there for different theories are there, 3 hours, 4 hours, and all that. So fomite, but likelihood is less. Then another one is the last one is aerosol, which is being talked about recently more. That what it says is the virus containing body excretions. Could be what are excretions? Body excretions, even excreta. Your excreta can be aerosolized into infectious virus containing droplets through a variety of ways. While talking, coughing, sneezing, they become a small particle, and then it will be the aerosol floating. Even medical procedures like bronchoscopy, tracheotomy, or even toilet flushing can lead to this kind of infection. That's why people say don't use the same toilet. Once a person uses, infected person uses, you need to clean it or sterilize it, then you use it. These infections are possible through this mode of, but reasonably less, but it's a possibility. So this is how it works. And why we call it zoonotic? Because this is going from an animal to human being. So such a virus or disease is called a zoonotic disease. Now look at the signs and symptoms of COVID-19. A symptomatic person has no symptoms, so no issue there. Then common mild symptoms are fever, cough, shortness of breath. Complications or emergency, you find pneumonia, acute respiratory syndrome. Symptoms are difficulty breathing, persistent chest pain, pressure, confusion, difficulty walking, bluish face, lips, all these are possible. Other symptoms are muscle pain, sputum production, diarrhea, sore throat, loss of smell. This can happen. This is called as anosmia, because you lose your, one of the symptoms is you lose your smell. So you will be very careful with that. And abdominal pains, nausea, vomiting, palpitations. Less common symptoms are upper respiratory symptoms such as sneezing, running nose. So these are common reported symptoms of COVID-19 if you're infected. Now let us look at what is a virus. A virus isn't a live in a typical sense because it doesn't need to eat or drink or breathe. It's a collection of genetic material, DNA or RNA, but for a COVID it is an RNA only, and a small toolbox of proteins contained in a shell called capsid. So its virus is nothing but a shell. Within that you've got a RNA, a genetic material which has got the codes for multiplication. Then you have a protein with that, which carries all the instructions for further. Once it invades the host system, this protein starts multiplying, and replications are possible because of the coded in that. So a virus uses its protein to perform two critical tasks. First one is get inside the cell of animal host, hijack the cell's own genetic material, the machinery. That's a very important thing. Then produce thousands and thousands of copies of it. So it's something like the virus jumps into the cell's internal printer, selects millions on the number of copies, and then hits the print button, and you get thousands and millions of viruses are being produced and then just thrown out. It's very dangerous. So this is the model of a viron of a coronavirus. Viron means it's a complete particle of a virus. Viron is a complete virus particle that consists of RNA or DNA with a protein coat. It's always have a coat that keeps it's intact. And these are the various kind of what you call proteins. You find structural proteins. This is the most important one, is called a spike protein, glycoprotein. That's why the name corona means crown-like. The spike protein, this is the one which the virus uses to connect to the host cell. Then you got other proteins like membrane protein, hemagglutinin envelope, then nucleoplastid, which is the RNA and the nucleoplastid protein, then small proteins, and there are some of them keep the structure intact. Some proteins are essential to see that the spike is able to connect well and that pretty stable. And this falls under the category of beta coronavirus, and the diameter is 120 nanometers. So this is what it looks like, and now we'll go into the somebody has raised a hand. Excuse me. Hello. Can you hear me? Hello.
Sir, yes, sir, we can hear you.
Raised a hand. I don't know.
Sir, yes, sir, we can hear you.
No. Somebody has raised a hand. I don't know why. Priya has raised a hand. I can continue.
Yeah. Please continue, sir. If they have any questions, we will do that in the end of the session.
Okay. 1 minute. Okay. Now we are coming to this is the mechanism how this coronavirus it enters and then it replicates. So before getting to the mechanism, I just wanted to quickly take you to the ACE2 receptor, which is very vital in that, because the moment the virus comes from the this is the alveoli, this is a coronavirus gets in the host, and from this in the alveoli you find a lot of ACE2 receptors, and the corona first binds to that, then it enters into the host via a process called as endocytosis, and then release the viral genome. So before getting into the detailed mechanism, I would love to take you to the ACE2. Certain things which are very important. So the SARS coronavirus binds to the ACE2 receptor using a spike-like protein on its surface. So it first binds to that, like a key being inserted into a lock prior to the entry and infection of the cells, and hence ACE2 acts as a cellular doorway. So ACE2 receptor is the cellular doorway with which the virus first connects, and for the virus to cause COVID-19. So ACE2 is a protein present on the surface of many cells, but it is abundantly found in the following organs. Like you find them abundantly in lungs, especially in alveoli. That's a lungs is first affected. Then you go to you find them in arteries, you find them in heart, you find them in kidney, liver, GIT. So wherever you find the expression of ACE2, coronavirus can attack them pretty easily, and you are sensitive to that. That's why these organs get affected. There are evidences which suggest that ACE2 may be higher in patients with hypertension, diabetes, coronary disease, etc. That's why we say that people with underlying symptoms like diabetics, hypertension, or cardiac patients should be much more careful, because they have more of ACE2, and the virus can attack them much more easier. So you are more prone to getting infected. And the density of ACE2 in each tissue correlates to the severity of the COVID-19 disease. That means whichever organ or tissue has got more density of ACE2, you can expect the COVID-19 to be more severe there. So this is a very important aspect there. And what is the role of ACE2? ACE2 helps to modulate many activities of the protein called as angiotensin 2 here, that increases the blood pressure, inflammation, increasing damage of blood vessels, linings, various types of tissue injury. So ACE2 will damage, that's a basic role. And what does ACE2 is? It is trying to block that. So one coronavirus sits on this receptor, ACE2 is not available to control the activities of angiotensin 2. That's called as ANG2 activity. When the ANG2 activity is higher, naturally damages all the cells and lungs and heart. So that's the issue here. So when SARS-CoV-2 virus binds to ACE2, it prevents ACE2 from performing its normal function to regulate ANG2 signaling. So when ANG2 signal is not controlled, there's more damage of all the organs connected to that. So thus the ACE2 action is inhibited, removing the brakes from ANG2 signaling and making more ANG2 available to injuries of tissues. Thus decreasing braking likely contributes to injury, especially to lungs and heart. So the sense is that as ACE2 is not available, the ANG2 signaling is much higher, and which will lead to a lot of tissue damage and all kind of things. So the key factor that determines severity of damage in patients with COVID-19 is abnormally high ANG2 activity. And so there are drugs which block this. What you do is, when a COVID patient, they try to give ACE inhibitors and ARB drugs to control the ANG2 activity, and they find that the ACE inhibitors and ARB blockers have shown to have a protective effect against COVID patients. So this is sometimes given along with the COVID treatment regime. So with that, let me just get into the mechanism slide. I think you will have 5 minutes time, then we'll get back to the presentation.
Researchers from China have used cryo-electron microscopy to show how SARS-CoV-2 infects humans. The study published in Science says the virus targets a type of receptor found on human cells in lungs, heart, kidneys, and intestines. Citing Kingston University microbiologist Mark Fielder, Sky News reports that the virus seems to attack two types of lung cells: goblet cells that coat the respiratory tract with mucus, and ciliated cells that usually filter out pathogens. A previous study published in Science found the virus spike protein has two receptor binding domains, or RBDs, facing downward and another facing upward. These allow the virus to bind with and invade human cells. The virus targets a human ACE2 receptor that has bonded with an amino acid transporter. This subtype of ACE2 structure has never been discovered before. The virus uses the spike protein's up RBD to penetrate the cell. The virus then dissolves its own protein shell and releases its RNA payload inside the cell, according to British Society for Immunology. A study in Frontiers in Microbiology says a coronavirus hijacks the cell's structure to reproduce. The viral RNA takes over the host cell's endoplasmic reticulum to replicate itself and to manufacture the protein parts to make new viruses. According to the Society for Immunology, the hijacked cell's Golgi bodies then package viral RNA and proteins in a viral protein shell. This leads to the creation of new viruses that leave the infected cell via the membrane. The study in Frontiers in Microbiology says coronavirus takeover imposes stress on the host cell. Cell death or apoptosis is the result when the infection overwhelms the host cell's ability to maintain homeostasis.
Yeah, we are back. I believe this video could have given you a picture of how the virus enters into the human system, and it multiplies. It's able to cheat the host genetic machinery and able to make multiple copies of viron. It enters here, then all these processes happening. Translation, then transcriptions, all these processes happening, and then finally the viral is being formed, and then it's taken out as a process called as exocytosis, and then it of course bursts out. So this is how it works, and so this is not our interest, because just to give you an idea how the virus acts upon, we are just showing this, and we'll get back to the second part of the presentation. So far we have done on, we have been talking about the overview, we have been talking on this, the coronavirus, the types, the whole timeline, biological analysis, transmission, and then finally of a video clip on the mechanisms. Now we'll get into the rationale, need for repurposing strategy, why chloroquine and hydroxychloroquine. So this section, the basic idea will be, I'll be presenting on these topics. Section 2 will be on, we are looking at the need for repurposing strategy and why we have chosen chloroquine and hydroxychloroquine. Then we'll be attempting to discuss the toxicity of racemic chloroquine and hydroxychloroquine, a major concern that exists. Then we'll move on to analyze the pharmacokinetic, pharmacodynamic, and toxicological profiles of both the drugs from a mirror image perspective. Then look for any superiority, if any, that exists between the two enantiomers. Then of course we will go for that. And just if the switch is there, then you can go into that and will be taken for clinical trials. Currently, people are working only on the racemic hydroxychloroquine or chloroquine. So this is how the sketch goes in, and then we'll just get into each of these. Rational and repurposing strategy, and this is required because no approved vaccines are available yet. Absence of known efficacious therapeutic agents, desperate need to find drugs for COVID-19. All this, made us to propose a repurposing strategy of older. Repurposing means your existing drug, which will be tried for a new indications, because chloroquine and hydroxychloroquine is known for hundreds of years. But then you are trying to use it for because of following reasons you are trying to do that. And in fact, this repurposing approach has been employed earlier to treat other viral infections such as Ebola outbreak in 2013. So this is not the first time that repurposing is being done. The most notable and also controversial repurposed drugs for COVID-19 is antimalarial drugs, chloroquine and hydroxychloroquine. Interestingly, if you see that, Donald Trump, in fact, is almost not selling exactly, he's touting on the, saying that he's using hydroxychloroquine. Hydroxychloroquine, 1 minute, I'm not able to hide this. This is coming up again and again. Okay. Using hydroxychloroquine is a prophylactic, prophylactic way. So Trump was talking, the man who's standing here is the director of the National Institute of Allergy and Infections. And so indirectly, Trump was, recommending that, hydroxychloroquine, and taken along with azithromycin. Could be a preventive approach. Then initial there are promising results are being reported when combined with azithromycin and zinc supplements. Then why did we choose chloroquine and hydroxychloroquine? There are the capabilities, because the immunomodulatory activity exerted by chloroquine and hydroxychloroquine suggested their potential use in the treatment of human infections, especially bacterial, fungal, viral. So we thought it can be used. Again, these drugs are reported to have antiviral effects for a wide range of viruses, especially SARS, MERS, Ebola, HIV infections, etc. Then the capability of chloroquine to inhibit replications of coronaviruses, a large family of RNA viruses that usually cause mild to moderate upper respiratory tract infection. So these are the data which we have. All these made them to think that yes, there's a possibility that we can try on this. Now you look at this again, chloroquine and hydroxychloroquine have been used for decades by millions of patients, and we have wealth of information about the adverse events is available. That means you know the full profile of the molecule, so it's not much risky to go ahead with that. Instead of working on an unknown molecule, we have a full wealth of information about the adverse events which these molecules could create. So that is one reason why we've chosen that. Then of course, conveniently can be given an oral tablets against your remdesivir is another candidate because that should be given by IV and it's much more expensive. So chloroquine that way is oral tablets and it's the least expensive drug being considered. So that will be very important from a global population should be able to afford. So chloroquine will be a better, and hydroxychloroquine could be a better candidate from the point of view of expensive price as well. Besides, US FDA has granted an emergency use authorization. You call it as EUA for both the drugs for treating COVID-19 because in emergency situations, these FDA Food Drugs Administrations permit you to go ahead with it urgently when you have a situation like this. Similarly, even remdesivir is another repurposed antiviral drug that has been given EUA for treating COVID-19. So currently there are about 141 and 296 ongoing or planned clinical trials. You can check the clinicaltrials.gov against COVID-19 using chloroquine and hydroxychloroquine. So a lot of works are going on. So with the above background, it makes sense in a situation of public health emergency and the absence of any known efficient therapy to investigate the possible effects of chloroquine and hydroxychloroquine against COVID-19. And further, there are three mechanisms being proposed, by which the chloroquine, and the hydroxychloroquine can, be effective against SARS-CoV-2. These are the sites where it's believed to act upon, and they believe that one mechanism is that they can hamper the virus receptor binding. Then they increase the pH of acidic cellular organelles, hindering endocytosis, and so it will hinder the endocytosis process. Then lastly, they can counter the process of viral assembly and viral protein synthesis. So all these, the three methods or mechanisms are proposed, and they assume this is possible. So with all this background, they thought that repurposing and using these drugs are worthwhile. So having said that, now just look into some of the facts. One of the major, concerns with how do I close this? I don't know. It's moving. How do I hide this? I don't know. Okay. The toxicity, because the toxicity profile of chloroquine and hydroxychloroquine have been well known for many years. Their administration is safe, although both can have serious side effects, especially at high doses and when combined with other medicines. So the basic concern is that they are believed to cause cardiac toxicity and retinopathy, the eyes, retinopathy. These are the two, toxicity which people, claim on this. That's the reason why they are afraid to get into this. In fact, there's a couple of papers on this, and if you look at this Lancet and the New England Journal of Medicine, what they have done is, there are reports, but these papers are withdrawn later. But initially, they claimed that there's substantial increase deaths and incidence of heartbeat rhythm changes associated with the use of hydroxychloroquine and closely related drug chloroquine. So they said that because of that, a lot of deaths are happening, and this paper was published in Lancet and New England Journal of Medicine, very reputed journals in USA. Then after that, immediately 2 editors and more than 1,000 scientists from across the world flagged major discrepancies in the quality of the data used by the two studies. It means, they questioned the validity of the data, and then later this paper was retracted from Lancet and New England Journal of Medicine. See, this was retracted later. Both the papers are withdrawn, and they said that then who said that the clinical trials on hydroxychloroquine will continue. So the drug is back again. Chloroquine is a moderate inhibitor of what is called as HERG. HERG is a potassium channel are essential for normal electrical activity in the heart. When you block HERG is this human ether ego related gene. which has got an encoded potassium ion channel and plays a major role in the cardiac repolarization. So when this is blocked, naturally you are bound to get some cardiac issues. That's why people are talking about this toxicity. And there are a number of clinical successful drugs in the market had a tendency to inhibit HERG and lengthening the QT intervals, potentially leading to fatal irregularity of the heartbeat. So the racemic has got this activity. Now the question is, we'll try to investigate on the both the enantiomers and see whether both of them have got the same kind of profile. So that's the next issue there. Coming into the chloroquine, hydroxychloroquine through the looking class, these two drugs are administered as racemates, and for many years, including the approval of chloroquine and hydroxychloroquine, pharmacopoeia were dominated with racemates. You have only a mixture of both enantiomers. But the trend was inverted in 1990 when new molecular entities, drugs were developed and marketed as single enantiomers. You call them as enantiopure drugs. So before getting into the chiral switch strategy, I would like to just take you to the one or two slides on the impact of chirality on biological activity, because I find a lot of students are out there, and many of you might be not familiar with how does chiral molecules behave in a biological environment in human system. So I thought it'll be nice to take a couple of slides on that and exactly tell you how the enantiomers differ in their pharmacokinetic and pharmacodynamic profiles. So this is a simple definition, and as you have an object here, there's a mirror image. You cannot superimpose. You refer such pairs as enantiomers, chiral twins, or handed molecules, or mirror image molecules, and they differ in their behaviors. We'll see that now. So the behavior of a chiral molecule depends upon the environment in which they are present. If they are present in a chiral environment, their behavior is totally different, and their behavior is more pronounced in a chiral environment. So when a chiral molecule is present in a chiral environment, they behave in a different way. For example, interestingly, biological systems and human body are highly chiral at a molecular level. They are filled with chiral discriminators. We are filled with carbohydrates, proteins, amino acids. All these are chiral. So human system can differentiate left-handed molecules from right-handed molecules. In other words, they can act upon them differently. So it becomes, so that's a very interesting case. And I'll give a simple example to make you understand that. Limonene is a simple molecule with one chiral center. This can exist in two enantiomeric forms. One form is called the R-limonene, other is called the S-limonene. R-limonene has got an orange smell. S-limonene has got an orange smell. These are two different, properties. And what I'm doing here is they are called as mirrored configuration. I'm changing shape of the molecule. So when I change from R to S, I'm changing the shape of the molecule. And because of changing the shape, it's believed that one of the molecule in that shape is able to trigger a receptor which is responsible for orange smell, and the other molecule in that shape is triggering a receptor responsible for orange lemon smell, which means that a change in shape gives you widely differing biological properties. Then you think about biological drugs and other issues, which could be very complex. We'll see that. Let us now look at the sequence of biological actions and how human body may handle chiral twins. This is the sequence of biological actions. You're all familiar. You've got a racemic drug here. There's an oral drug given. There's a disintegration, dissolution happening. Then you have pharmaceutical phase. What survives after that is called pharmaceutical availability. Gets into the pharmacokinetic phase, where you've got absorption, distribution, metabolism, and elimination. Then what survives is called bioavailability or biological availability. And then enters into the pharmacodynamic phase, where you've got a ligand-protein interaction. Then you get a response, which could be toxic or effect. That's not an issue. The issue here is, by definition, racemic mixtures are 50-50. But the moment you put them into a biological system, it is no more 50-50, because all the phases, pharmaceutical phase, pharmacokinetic phase, pharmacodynamic phase can be enantiomer selective, which means that one enantiomer may be protein bound, other will not be protein bound. One enantiomer may be eliminated, other will not be eliminated. One may be what you call metabolized, other may not be metabolized. So you find that it is never 50-50 in vivo. It will be varying 60-20. We don't know. We have to analyze. That's why you need to track them separately. So the point, we'll give one or two examples to tell you how these kinetic molecules behave. Salbutamol is a molecule with a chiral center. This is the R-salbutamol. This is S-salbutamol. This is a eutomer, the desired activity, bronchodilator. So you find here there's a stereoselective metabolism. The R enantiomer get metabolized, whereas the S is not getting metabolized. But R is the desired molecule, and when the R is removed from the biological system or from the blood, there's a drop in the bioavailability. This is how enantiomers behave. Enantioselective, pharmacokinetic, number of drugs. The point here is, whenever you have a chiral molecule, you need to look at them differently. Otherwise, you will miss these important aspects. One more drug here. This is dextrabiprazole. This is rivorabiprazole. Look at those kinetic parameters. All of them are different. You look Cmax, 576. Here is 234. Area under the curve, 1972, 460. T-half, 2.74, 1.5. You find that pharmacokinetic parameters are totally different. So you cannot, that why I'm telling you, what I'm trying to tell you is that similarly, you need to look at chloroquine and hydroxychloroquine. They have not done studies on that. These enantiomers, you are telling the racemic chloroquine is toxic, but maybe you separate them, one is not toxic. In that case, you can use that enantiomer. That's the point. So here, this is the kinetic, and now let's look at the pharmacodynamic, a few examples here. Look at ibuprofen. Ibuprofen, the S enantiomer is 100 times more active than the other SR enantiomer, distomer. The enantiomer, the desired active is called eutomer, other one is called distomer. Okay. For example, look at penicillamine. It is treatment used in the treatment of Wilson's disease, yes. And whereas the R enantiomer has got a toxic effect. Not okay. So yeah, widely different. This is an interesting case. Look at propoxyphene. Propoxyphene is it's a pair of enantiomers. The one on the left-hand side, you call them as levopropoxyphene. You call this as a dextropropoxyphene. This has got antitussive action. This has got analgesic action. So both of them enantiomers to different therapeutic indications. Very, very interesting. Whereas one has got antitussive, other has got analgesic. These are separated and marketed as two different products by Eli Lilly company. This is called as Novrad, and the name here is, you look at the reflection here, Davron. So what I'm trying to say is enantiomers can have widely differing spectrum of activities. So it's very important to understand that. Few more examples here. You can look at that, verapamil and the S is 10 to 20 times more active and hypertensive. Timolol is it's an angina here, but this has got a glaucoma side effect. Warfarin is 2 to 5 times more potent than the other enantiomer. Salbutamol R is 68 times more potent than the other enantiomer. So the point here is, in biological system, racemic molecules, the enantiomers behave widely differently. So there's always need of a chiral perspective if the molecule under question is chiral. So our molecule here, which we have taken up, is chloroquine and hydroxychloroquine. Both are racemic drugs. So the next part, I'll be going to that, and we'll be looking at the separate properties of these enantiomers and trying to say that which one will be better and can we use it for a potential drug as in the treatment of COVID or as a prophylactic drug. So now let's see what are these chiral switches. Chiral switches are racemic drugs which have been switched to either a levo or the levo form, the dextro form, into a single enantiomer for a perceived improved therapeutic efficacy, safety, or to manage a life cycle to extend the patent. So this is all. You have a racemic therapeutic already available. So for example, levocitrazine is a racemic drug. You switch to citrazine is a racemic drug. You switch to levocitrazine. This is a pure enantiomer. That's how it works. So you do that. These are not treated as inventions, but you are treat them as selection inventions. I'll give you one approach here. See, this is ibuprofen racemic drug. I switch to a single enantiomer. So this is called as an enantiomer drug. And this is called chiral switch. Sometimes you call them as a racemic switch. But to perform these switches, you need to what is called as you have to perform bridging studies. But in the case of COVID-19, I believe bridging studies have been given a little leniency. They can quickly explore on these because to get a useful drug. So this is, you call them as a chiral switch. I'll give you some more examples here so that you make it interesting. All these drugs are in the market today. From cardiology, amlodipine. It's an enantiomeric switch of amlodipine. Atenolol, racemic atenolol, switch of that. From gastroenterology area, you find esomeprazole. It's a proton pump. You got pantoprazole, the pure enantiomer switch is called S-pantop. So you find number of drugs are in the market. See, ofloxacin, which is levofloxacin. So many more drugs. I'm not going to go this because that will be consuming a lot of time. But all these are available, and these are the drugs in Indian market. These drugs are in Indian market. All these drugs, S-amlodipine, S-metoprolol, S-pantoprazole. These you call them as unichiral drugs. And the company maximum involved in chiral research here is MCure from Pune. They manufacture all these drugs. And these are the other companies which are engaged in chiral research. So a lot of India is capable of carrying out these kind of researches. Dr. Reddy's, Cipla, then you got Cyrus, MCure is a leading company, Torrent, and all these companies. Okay. Having said that, now let's look at now we'll be looking at from a chiral. So far we have been talking about racemic drugs and how the enantiomers are different. Now we'll get into how these chloroquine and hydroxychloroquine enantiomers will be behaving. And the rest of the papers are based on a couple of publications which has been done in 2020. One is published in Molecules, another is published in Drug Discovery, another one is from China. Not yet, it's in the pre-publication stages. So the references are from there. See, this is R chloroquine. You could say chiral center. The mirror image is S chloroquine, chiral center here. This is R, this is S. This is hydroxychloroquine. This is the chiral center R here. This is S. So these two are mirror images. So this is the structure. How does it look like? Now let's see. I'm not able to hide this. How to hide this? How can I hide this? This is coming down every time. What should I do about it? I don't know. Okay. The facts on the chloroquine and hydroxychloroquine through the looking glass. So the molecular blocking studies on the HERG channel, because we said HERG is blocking, is giving you a toxicity. So molecular blocking studies on HERG channel blockade suggest that the chloroquine mainly interacts through 3-point attachment model, which means that these are the 3 points which are being mentioned here. These are the amino acid residues. And when I say 3-point attachment model, chloroquine is something like this. It's called as Eason treatment model, 3-point attachment model. You have a drug molecule here, and the 3 functional groups, those amino acid residues will be interacting with them. Here you find the 3-point attachments. You call this as a good fit, and possibly, and here you find a weaker interaction. There's only 1 point attaching. So this is normally the eutomer, and this will be the distomer, which will give you the desired activity. So when I have a toxicity, maybe one of them is interacting strongly, is producing a toxic effect. The other one, which is interacting weakerly, may not have so much of toxicity, means which can be less toxic. So maybe among this, you can use this enantiomer. That's a point here. Come back here, and then so it may be anticipated that the chloroquine off-target activity on cardiac functioning could display stereoselectivity. So the 3D requirements might cause stereoselectivity of blocks, and one of the chloroquine enantiomers might display weaker interactions with respect to its chiral twin. So one of them will be, giving you a toxic effect, whereas the other one will not be that toxic. In that case, you can separate and use them. That's idea. Thus resulting in a less harmful effect on cardiac function. So this is a new approach. The racemic drug has toxicity, but the enantiomers, there's a possibility that you can switch. So this means that one of the enantiomers of chloroquine could be less active as a HERG blocker, and the cardiac toxicity may be less or may not be there, and consequently safer to humans. Another side effect which has been repeatedly concerning is the severe side effect of hydroxychloroquine is retinopathy, caused by the enantioselective accumulation of R enantiomer and not the S enantiomer, which means that the R enantiomer is getting into the ocular tissue and giving you a toxic effect, whereas yes, hydroxychloroquine is not toxic, which means that then there is a possibility that you can look in for S and separate and use where the toxicity will be less or may not be there. So that's the point I'm trying to tell you. So that's indicating the possibility of a chiral switch of chloroquine or hydroxychloroquine. Chloroquine has got cardiac toxicity and hydroxychloroquine has got retinopathy. And both of them, the enantiomers, one of the enantiomers are showing possibly less toxicity, and we can explore that. Okay. Now let us the facts on the chiral profile, the pharmacokinetic and dynamic properties of both the drugs. We're looking at antiviral activities. In vitro studies have been done on the racemic, enantiomeric chloroquine and hydroxychloroquine. If you look at here, chloroquine racemic is this is the configuration, optical rotations, and the IC50 value. Inhibition concentration 50 in micromoles, and you find that racemic requires a higher dose, 1.8, and R requires 1.975 micromoles, whereas this enantiomer is requiring a lower dose, 1.761. Whereas if you look at hydroxychloroquine, you find that the value is still less than half, 1.44 micromoles, which means that, for example, azithromycin requires almost more than 10, 14.57, which means that the enantiomers are much more requiring lower dose, indicating naturally a lower side effect. And among the hydroxychloroquine and chloroquine, you find that hydroxy has got much more, almost less than half, 1.4, which means that what this data suggests, this actual study done and published. So the data suggests that chloroquine and hydroxychloroquine exhibited more pronounced activities than their respective R enantiomer in the assay against SARS-CoV-2 in vitro. And chloroquine seems to be less toxic than the R. That's how it goes. Okay. Now look at the other the data which are on the kinetics and renal clearance. The S enantiomer is 2 times quickly renal clearance than the R isomer, which means that there is toxicity could be less and the shorter elimination half-life. And the protein binding, if you look at the R hydroxychloroquine, is got 37%, whereas the S hydroxychloroquine 64%, meaning that the R has got a wider volume of distribution. So that's why the R possibly is entering into the ocular tissues and causing toxicity of the cardiac toxicity, whereas the S binding is more, so the distribution to the ocular tissue is much less. So this is all in vitro assay done on in human plasma. So these data are there indicating the possibility for a switch. Further studies suggest that there is no inversion between S-hydroxychloroquine and R-chloroxychloroquine. This means that there is a tendency called as many of the enantiomers, S can switch to R can chiral inversion. If there's a possibility for such inversions because of the lack of stability of the chiral center, such molecules cannot be considered for chiral switches because whichever way you switch, it will racemize. So whereas you find that the studies indicate that this hydroxychloroquine is pretty stable, so there is no question of chiral inversion. So you can straight away going for a chiral switch. I'll just get a point here. For example, here this is a thalidomide molecule, which you all know. This is a racemic thalidomide, and if you separate them to a switch like R switch, it is observed that in vivo, in human system, NAR thalidomide has a tendency to invert it to the S form, which means that whichever form you separate, in the in vivo, it will get into racemize. This is called as chiral inversion, and such switches will not work, and we cannot do that. So both the drugs, in chloroquine and hydroxychloroquine will be safely can be used as a switch. I think I moved in a little. Sorry, I just jumped the track. 1 minute. I'll just get rid of that. Sorry, I just link. I'll get back to that. I missed the link somewhere. Switches here. Yeah, we are here. Sorry, a lot of hyperlinks out there. Yeah, we have been talking about that. Yes. So this is qualifying for a switch here. Okay. Now we're going to the last part of it and then how develop the promising enantiomer. So we saw that all the studies demonstrate enantiomer selective antiviral activities of chloroquine and hydroxychloroquine against SARS-CoV-2. The S enantiomer of both chloroquine and hydroxychloroquine proved to be, the S enantiomer of chloroquine is 27% active, whereas the hydroxychloroquine is somewhere around 60% more active than their corresponding enantiomer. So in both the cases, you find that yes, enantiomer is much more active. So there's a possibility among these two, you can always try with the higher activity, 60% of the S hydroxychloroquine. So in vitro antiviral activity is mainly derived from the S isomer. It is reasonable to expect a drug dose can be reduced to half of that or even less than the estimates for a similar antiviral effect, eliminating the side effects of the distomer R isomer. So from all this data, there's a possibility for a developing chiral switch of the safer enantiomer, the S enantiomer of the hydroxychloroquine, based on the following expectations: safer pharmacological profiles of utomar, favorable risk-benefit profiles, improved therapeutic index, shortened development and approval process. So there's a quite quick possibility for a chiral switch, and of course, further clinical studies should be employed using the S-hydroxychloroquine and the chiral switch, potentially superior drug for the treatment of COVID-19. So even as on today, most of the studies are being done on racemic therapeutics. What we are trying to suggest is that all the clinical trials in future should be done on pure enantiomers like a switch, which is much more favorable. And in fact, hydroxychloroquine racemate is available plenty in the worldwide of more than 3,300 metric tons. So raw material is not an issue. You can separate racemic mixture into pure enantiomers easily by industrial scales by simulated moving blood technology or preparative chiral chromatography. Separation is not an issue. So people should not tell that we are not able to separate. Then you can approach asymmetric synthesis is available for separations. So there is no excuse to continue running clinical trials using racemic chloroquine or racemic hydroxychloroquine. Questions, comments? So if the molecule under study is stereoisomeric, in particular chiral, then all the investigations need to be done from a mirror image perspective, else the data generated will be meaningless or nonsense. Which meaning to say that, see, for example, if you're working on a chiral molecule, like for example, chloroquine or hydroxychloroquine, and if it's a racemic, you need to perform studies on the both the enantiomers and look for those data. Otherwise, the data generated will be meaningless and nonsense. We live in a three-dimensional world, and we should insist on a three-dimensional science. If any questions, I'll be glad to answer. I believe I've been able to convince you. My only point here is that if the molecule under question is chiral, then you need to have a mirror image perspective. That's very, very important. Otherwise, the data generated will be of no use. And these are some slides to prevent the spread of COVID-19 in community. You're all familiar. Know your spread. Stay home if you're sick. Cover, cough, and all your social distancing. All these are there. So I think I've done with my presentation, and Carol, yours. Thank you.
Thank you, sir, for your wonderful presentation.
I believe I've done my job. Yes.
It's very nice. I hope our participants have gained knowledge about the chiral switch, importance of chirality, and how the enantiomers they are having various applications in today's world, and how the one enantiomer of a chiral drug may be medicine for a disease in another enantiomer can be a toxic effect. So it was nice presentation, sir. Thank you. We have one question in chat box. So can I have that question?
Yeah, sure, sure. Can I stop the screen sharing or should I keep it on?
So you can stop. And I request other participants to post the questions. I can read out for you. The first question is, can human invent a new virus? Corona invention is a naturally occurred or man-made?
That's a debate. People say it has been, created, and I think the questioner, if he very clearly knows, I put one, disclaimer on the first slide, where I say that I am not a virologist or I'm not a microbiologist. So there are a lot of rumors, saying that, where this virus is created in China and man-made and all that. I'm not an authority to say on that. So that's why I know such questions will come up, and I'm not an authority on that. But there are, you because of all these YouTubes and all kind of available. There are concerns about that. People believe it has been man-made and all that. I'm not sure about that.
Thank you, sir. One more question is there, sir. Comparatively, which one is better for clinical management of patients? Chloroquine or hydroxychloroquine, or some antiviral like remdesivir, or some corticosteroids like methylprednisolone?
They normally do it on a what do you call it? Not a single drug is being given. In fact, nowadays people are using remdesivir as an antiviral. Chloroquine has got, but among chloroquine, hydroxychloroquine will be better because of the safety profiles. And of course, people are using that. I believe there are people taking it is taken as a prophylactic, dose. Not that people who are believed to be in touch with the patients continuously. Such people, they say doctors do take hydroxychloroquine and along with the azithromycin. So I think it's not a single drug to comment on that. Yes.
Thank you, sir. Only we had, sir, 2 questions. Thank you very much, sir, for the clarification of the doubts. Before we end the session, I would like to thank the speaker of the day, Dr. Valiyappan Kanappan, sir, to accept our invitation in his busy schedule. I would like to thank the Dean, management, and the staff of College of Pharmaceutical Sciences, Dhanand Sagar University, for giving me an opportunity to conduct this webinar. I thank the promotion team, Ms. Deepa, Mr. Shakat Ali, Mr. Hemant Kumar, Mr. Mohammad Imtiaz for their continuous support till the end. I also thank all the participants who attended the session and made the session successful. Thank you all.
Thank you so much.
Thank you, sir.
I hope
Participants can fill up the feedback form posted in the chat box. You'll receive the certificate soon.
Yeah, please let me know the feedback. I always love to know that and what I should be doing it in my further presentations and all that. Right? Yeah. Okay, thank you. Nice. Thank you so much.
Thank you, sir.
Can I leave now?
Yes, sir.